This video is going to describe the process of bacterial conjugation. So again, this is one of the three mechanisms of horizontal gene transfer that bacteria use to acquire new pieces of DNA and therefore potentially acquire new characteristics or traits. So horizontal gene transfer always involves a donor cell. So we have a donor cell over to the left, and then we also have a recipient cell. So what's important about conjugation is there's going to be a structure that forms between these two called a pilus that's going to be the structure that allows DNA to move from the donor to the recipient. And what we have at the beginning of this is in this donor cell, there's two pieces of DNA we want to focus on. As we have said many, many times, all cells are going to have the chromosomes that are necessary for supporting all the processes that a cell needs, an organism needs to be successful. And so that DNA in the form of chromosomes is always going to be present within cells. Something that can be present in bacteria are called plasmids. So these are additional pieces of DNA. So this is double-stranded. That's what these two circles are supposed to represent. But it's separate from the chromosome. And plasmids are always going to contain non-essential characteristics, non-essential information. So another way of trying to understand that is if I could go in with a pair of tweezers and pluck out this plasmid, there still is all the information needed for that bacterial cell to survive totally fine. If we were to do the same thing and take a little pair of molecular tweezers and remove the chromosome, that would be removing critical essential information that the cell needs, and the cell would die. Okay, so although there are these two pieces of DNA, the chromosome and the plasmid, bacterial chromosomes have the essential information. Plasmids are going to have non-essential information. Now, this particular plasmid is called the fertility plasmid. And fertility plasmids, the non-essential information they have is the ability to conjugate. And so this is why this cell has been designated an F+ cell. It has a fertility plasmid. And notice over this recipient cell, while it does have the essential chromosomal DNA, this lacks a plasmid, a fertility plasmid at this point. And so these are designated F+ because it has the fertility plasmid. This is F-, it lacks the plasmid. And so it's going to be this F+ cell because it has this extra information, this non-essential information that enables them to initiate this process of conjugation. And so one bit of information on this plasmid is to make a pilus. So this pilus is basically a tube that connects these two cells, and the F+ cell is going to donate some DNA. And what you're going to see is one strand, so one of these circles is going to break apart and move through this pilus. And so this recipient cell is going to get this one strand, and it's going to become a circle. And so what you see now is a single strand in the recipient of the plasmid, a single strand in the donor, but both of these cells have the ability to, based on the single strand DNA that's present, make a complementary second strand. And so what's going to happen is this cell is going to restore its double-stranded fertility plasmid. And when this one strand is used to make a second strand, you can now see that this cell has something it didn't have before. It has a copy of the fertility plasmid. And so while it was an F- cell at the beginning, it is going to convert to an F+ cell because, as we said at the beginning, what makes something an F+ cell is it has the fertility plasmid, and this indeed does now have that. Now, one other thing I should mention is the minimum amount of information on a fertility plasmid is how to conjugate, right? How to build a pilus, how to donate this DNA. But there can be other non-essential information within that fertility plasmid as well. So we know that antibiotic resistance genes can frequently be passed with these fertility plasmids. There can be other disease-causing genes, we call them virulence genes, that can be present there as well. And so we don't know everything that's on this plasmid, but we know there is the information on how to conjugate, and there can be other information that would be beneficial for this recipient cell as well. So that is a quick video on bacterial conjugation. This specifically is looking at an F plus to an F minus. Just realize there's another type of conjugation that uses a special F plus cell known as an HFR cell, and we'll talk about that in another video.